Hello everyone, myself Manasi Rajan Korgaonkar. I am going to explain about a regulatory mechanism involved in eukaryotic cells. So first, let us understand what is gene regulation. So gene regulation is the process used to control the timing, location and amount in which genes are expressed. The process is carried out by a variety of mechanisms including through regulatory proteins and chemical modifications of DNA. So gene regulation is the process of controlling which genes in a cell's DNA are expressed that is used to make a functional product such as protein. So regulation of gene expression can take place at any levels such as DNA is transcribed into mRNA and mRNA is translated into protein. So these are basically 5 levels: epigenetic, transcriptional, post-transcriptional, translational and post-translational. So here this is the diagram of central dogma, and these arrows indicate where each type of eukaryotic regulation of gene expression interrupts it. As transcription serves as a regulatory point in the first phase of gene expression, therefore, let us know about one such mechanism, that is functional on transcriptional level, that is CRISPR mediated modular RNA guided regulation. The usual CRISPR system means clustered regularly interspaced short palindromic repeat system is used to regulate gene expression. CRISPR can be repurposed to function as an RNA-guided DNA binding system to interfere with transcription of an arbitrary gene. So this system is known as CRISPR interference. So this is a RNA-based method for highly specific silencing of the transcription in prokaryotic or eukaryotic cells. So the typical CRISPR interference system is a type 2 CRISPR machinery of Streptococcus pyogenes. Using this, we can either activate or repress gene expression. So the basic purpose of this CRISPR interference is regulation instead of modification. So in this picture, as you can see, there are the factors which are included in CRISPR interference. First is the sgRNA, which is a single guide RNA. Second is the D-Cas9 protein, and third are the transcriptional effectors, which can either be repressors or activators. So let us understand the mechanism of CRISPR interference system. The usual CRISPR system is composed of two components, a guide RNA and a CRISPR associated endonuclease enzyme, which is Cas9. But here the CRISPR interference system consists of single guide RNA and catalytically dead or inactive Cas9, which is D-Cas9 enzyme, which lacking endonuclease activity. The guide RNA is complementary to the region of the gene of interest, which is to be repressed or activated. D-Cas9 is also known as endonuclease deficient Cas9 because it is a mutant form of Cas9 whose endonuclease activity is removed by mutating the endonuclease domains. Single guide RNA is a single RNA molecule which consists of both CRISPR RNA and tracer RNA. So let us understand the mechanism with the help of this diagram. D-Cas9 guided by single guide RNA forms a D-Cas9 single guide RNA complex, as you can see in this picture. So the D-Cas9 can bind to the target DNA based on the guide RNA targeting sequence, but it cannot cleave the target DNA. So by targeting gene at the promoter, the complex is paired with a transcriptional effector, which can either be a repressor or activator. So example of repressor, as you can see here, is D-Cas9 CRAB. An example of activator is D-Cas9 PP64. So these effectors interfere with RNA polymerase. So without binding to RNA polymerase and transcriptional factors, the transcription of target gene is inhibited. So the CRISPR interference basically represses transcription by blocking either transcription initiation or transcriptional elongation. So this picture explains about it, how CRISPR interference represses the transcriptional initiation and transcriptional elongation. So this repression is strand specific and it is stronger due to activity of helicase, which unwinds RNA DNA heteroduplex ahead of RNA polymerase II when the single guide RNA is complemented to the template strand. One of the examples in eukaryotes where CRISPR interference is used is that it can also repress transcription via an effector domain. So fusing a repressor domain to D-Cas9 allows transcription to be further repressed by inducing heterochromatinization, which means conversion of euchromatin to heterochromatin. So green fluorescent proteins can also be used as fluorescent markers. For example, the couple associated box KRAB domain can be fused to D-Cas9 to repress transcription of the target gene up to 99% in human cells. So this is the example in this picture how CRAB is fused with D-Cas9. So these are some applications of CRISPR interference. CRISPR interference is highly applicable in retinal diseases. It is also used in vivo knockdown of target genes and ex vivo knockdown of target genes. So in vivo knockdown, CRISPR interference has been used to treat familial hypercholesterolemia and obesity in mouse models in vivo by knocking down genes that are involved in the disease pathology. And in ex vivo knockdown, CRISPR interference is applied to cells which are then transplanted into a patient. So these are the references which are used for this presentation. So I hope this presentation has helped you to learn about CRISPR interference as regulatory mechanism in eukaryotic cells. Thank you.